Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing IIT JAM question paper Biotechnology for the year 2019. This is the numerical aptitude test that I am discussing here. If you want still previous year question papers of IIT JAM Biotechnology being solved, you can go to my channel Omni Bio Entrance. See for in the playlist C for IIT JAM, there you will find. If you are new to my channel, please do like, share and subscribe. Coming to the first question. C3 plants utilize 18 molecules of ATP to synthesize 1 molecule of glucose from carbon dioxide. Okay. How many molecules of ATP equivalents are used by C4 plants to synthesize 1 molecule of glucose from carbon dioxide? So glucose is a 6-carbon compound. So in the question they have given C3 plant utilize 18 molecules of ATP to synthesize 1 molecule of glucose. So from carbon dioxide. Now 6-carbon compound. So carbon dioxide also will be more in number. Now C3 plants require 3 ATP for 1 carbon dioxide fixation because it is 18 molecules for 6 carbon of glucose. So 1 carbon of glucose it will require 3 ATP molecules. Now C4 plants they require 5 ATP for 1 carbon dioxide fixation. So for 6 carbon molecules of glucose to get fixed, it requires 6 5s are 30 ATP molecules. Then next one. A 0.1% weight by volume solution of a protein absorbs 20% of the incident light. What fraction of light is transmitted if the concentration is increased to 0.4%? So there is a formula from Beer-Lambert's log I0 by I is equal to epsilon Cl, where I0 is intensity of incident light, I is intensity of transmitted light. Epsilon is molar absorption coefficient, C is the concentration, L is the path length. Now 20% of the light is absorbed means 100 minus 20, that is 80% of the light is transmitted. Okay. So log 100 by 80, that is I0 by I is equal to epsilon into 0.1. A 0.1 weight by volume solution of protein absorbs 20% of incident light is given. So concentration of protein is 0.1, path length is the same 1 centimeter. So from there you will get the value of epsilon, that is 0.969. Now new concentration is 0.4%. So transmitted light I will be then, that is what you have to find out. So log 100 by I is equal to epsilon Cl. Epsilon value you already got, it is 0.969 into new concentration is 0.4. So log 100 minus log I is equal to 0.3876. Calculate and you will get I as 40.92, which is 40%. Then next one. Heterozygous female fruit flies with grey body and purple eyes were mated with homozygous males with black body and red eyes. The number of offsprings obtained and their phenotypes are shown below. Calculate the recombinant frequency. So these are the number of offsprings, these are the phenotype. Now recombination frequency, it is given by the formula number of recombinants by total progeny into 100. So number of recombinants, that you have to see from the table. If you see the table, you can see that these values are more in number, whereas these values are less in number comparatively. So the one which is more in number, they will be your parentals. If you see the phenotype, it is grey body, purple eye for one parent and black body, red eyes for the other parent. So they are your parentals, whereas these 61 and 55, they are your recombinants. They will be lesser in number than the parentals. So number of recombinants will be 61 plus 55. And total progeny means you have to add all these 300 plus 347 plus 61 plus 55 into 100 and you will get it as 15.20. So that is the recombination frequency. Then next one. Proinsulin is an 84 residue polypeptide with 6 cysteines. How many different disulfide combinations are possible? So proinsulin is having 6 cysteines. So 1, 2, 3, 4, 5, 6. Disulfide bonds form between cysteines. So 1, 2, 2, 3, 3, 4, 4, 5 and 5, 6. So total, if you count all these, between 1 and 2, between 2 and 3, between 3 and 4, between 4 and 5, between 5 and 6. Then you will get between 1 and 3, 1 and 4, 1 and 5, 1 and 6. Then 1 and 2 is the same. So you will not, or 2 and 1 is the same. So you will not take that into consideration. You have to see these bonds between 2 and 3, 2 and 4, 2 and 5 and 2 and 6. Like that if you do and you count all these, you will get it as 15. Then next one. A schematic representation of restriction fragment length polymorphism analysis of a sample population is shown below. The number of people exhibiting a given pattern is indicated above the lanes. These are the number of people. These are the pattern. Calculate the frequency of 6.5 kb allele. So this is the 6.5 kb allele. You have to calculate the frequency. Now frequency calculation. So total number of people, if you add all these 80 plus 40 plus 20 plus 7 plus 13 plus 12, you will get it as 100. Now people exhibiting 6.5 kb allele, that is over these ones. 8, 20 and 13. So these are the people who are exhibiting the 6.5 kb allele. 8, 20 and 13. Add up 8, 20 and 13. Divide by total number of people. You will get the frequency as 0.41. Then next one. Phosphoglucoisomerase catalyzes the following reaction. That is glucose 6-phosphate to fructose 6-phosphate. A 0.5% of the original concentration of glucose 6-phosphate remains at equilibrium. Then the equilibrium constant of this reaction is. So equilibrium constant, it is given by the formula concentration of products by concentration of the reactant or the concentration of the substrate here. So here they have given the if the 0.05% is the original concentration of glucose 6-phosphate at equilibrium. Okay. So 100 will be the initial concentration of glucose 6-phosphate and 0.05% will be the one at equilibrium. Now what is the concentration of fructose 6-phosphate? Now if you see, we can take the concentration of fructose 6-phosphate as X at equilibrium. So initial minus final concentration of glucose 6-phosphate will give you the concentration of fructose 6-phosphate, and 100 minus 0.05 is 99.95. So equilibrium constant will be concentration of product by concentration of substrate, that is 1999. So that is the answer. Then next one. In a bacterium, a mutation resulted in an increase of Ks, that is substrate specific constant, for ammonium from 50 micromolar to 5000 micromolar without affecting the mu max. Mu max is maximum specific growth rate. The specific growth rate, that is mu of the mutant growing on 0.5 millimolar ammonium in the medium, decreases by a factor of. So here you know the formula, mu is equal to mu max into concentration of S by Ks plus S. So mu is specific growth rate, mu max is maximum specific growth rate, S is the substrate concentration. Here it is 0.5 millimolar. So that is 500 micromolar. Convert, because all the others are in micromolars. Okay. Then Ks is substrate specific rate constant. Initial Ks is 50 micromolar and final Ks is 5000 micromolar. Mu by mu max for initial, that is before mutation. It will be put in the formula. 500 by 50 plus 500, that is 0.9. Mu by mu max for mutant, that is 500 by 5000 plus 500, that is 0.09. Now divide 0.90 by 0.09, you will get it as 10. So it decreases by a factor of 10. Then next one. The total number of DNA molecule present after 5 cycles of polymerase chain reaction starting with 3 molecules of template DNA is. So it is given by the formula N0 into 2 raised to N, where N0 is the initial number of DNA templates and small n is the number of PCR cycles. So 3 into 2 raised to 5 will give you 96 as the answer. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you want still previous year question papers of IIT JAM been answered, you can go to my channel Omni Bio Entrance. See for IIT JAM in the playlist and there you will find them answered with explanations. Thank you.